his career. He joined in 1985, still managed to get a early career talk here. And he initially focused on the application of flow cytometry for the study of marine phytophytoplankton. And then he led the diversity of oceanic plankton team for many years, where he developed a broad research program that was focused on the diversity and ecology of eukaryotic phytophytoplankton, more specifically on green algae, but also other groups such as bolidophytes, aptophytes, that are involved in symbiosis, mainly with cyanobacteria. In 1998, he created the Roscoff culture collection, which under his steering has become the biggest or largest phytoplankton culture collection in the world. And he's also maintaining the PR2 database that he will be talking about today. So whenever you want, Daniel.
Okay, thank you very much. I'm going to share my screen first. If I can find it. Yeah, here. And I go for. Oops. Yeah, it's good. Everybody can see the screen? Yeah, thanks. Thanks, Andres, for the very nice introduction. Yeah, I feel like early career because when you are kind of visiting, you don't have so many things to do, so I can spend my time playing the things that I like to do. So today is going to be a bit more boring than the other talks that you have been listening to, because I'm going to talk about the database. But obviously from some of the talks that I heard, it's a database that have been used by many people, and I think it's quite useful. So I will just very briefly introduce metabarcoding, but you have seen several talks about metabarcoding, then talk about PR2, the status now, what are the use of the database, then introduce very shortly another database that I'm developing, which is called MetaPR2, which is a metabarcode database, and then what's next for PR2. So you have been introduced in many talks, like the one, for example, 2 talks today and some yesterday about metabarcoding, which is taking samples from any kind of environment, amplifying a universal gene, then assembling the data, putting them together, creating packs of sequences and assigning them to a taxonomy. And then you can go back to your environment and tell what are the different species present. There are many, of course, caveats with this approach based on the sample processing, based on the PCR primers that you use, based on the software that you use for processing the data. But one stumbling block is really the reference database, because if your reference database is bad, then there is no way that you can reconstruct the community. You may assign things that you think are diatoms to dinoflagellates and conversely. So the reference database is really critical. And people are using as gene, of course, 18S with 2 key regions, the V4 and the V9 region. But there are other markers that are used much less, like ITL, 16S plastid, RBCL. And as we saw yesterday, I think the next challenge, the next frontier will be to use operin, full operin, which provide a lot more resolution at the phylogenetic level, as Magnus showed yesterday. To do the assignment, if we just focus on this part, which is the one of the critical parts, is that you can use GenBank to assign your sequence, but the problem with GenBank is that taxonomy of the sequence is very bad. especially for environmental sequences, because the only information is environmental sequence. Okay, I know I got the sample from the environment, so I should get environmental sequences, but no more. Silva goes a bit further. It's quite good, I think, for prokaryotes, but for eukaryotes, it's not very, very good, actually. So this was the idea of developing the PR2 database, developing maybe a smaller database, but which with very high quality in terms of the annotation. So this is a long story. It came when Laure Guillou, who was doing her thesis in Roscoff, and then we assembled a big database at that point, which maybe had 5,000 sequence of 18S. During one project, the EU project called PICODIV, with people from Barcelona, Ramon Massana, people from Oslo, people from Germany, and people from UK, we developed a bigger database, which was using the ARB software. But this was still very clumsy to use. And then as part of the Biomax project in the around 2010, Laure Guillou decided to put this together and to assemble a better database. And with the help of Richard Christen, who was a bioinformatician in Nice, in the south of France, they put together the first release of the protein database, which at that time was online. You could download sequences using a web interface. It turned out that Richard was very close to retirement, and when he retired, the website just disappeared. He said, no, I don't have any more money to pay for the website, and from one day to the next, it was gone. So that's when I, with Laure and also Fabrice, not help, we decided to at least provide the community with some kind of reference database, but it was quite primitive, and it has evolved since then. Now we have a team around the PR2 database with a core team which is made of myself, Javier Del Campo, Laure Guillou, and also Fred May from the CIRAD, who has been developing a lot of bioinformatics software, especially the research suite. We have a scientific committee that help us to take decisions, to orientate the PR2 database. And more importantly, we have contributors. So some of them are attending now, who are helping with the annotation, because what's critical is to have a good annotation. So we rely on these contributors that provide us a list of sequence with a good taxonomy. For example, Bente, that you heard about today, in the talk of Miguel, she provided a very good database for Aptophyta. So the Aptophyta are very well annotated. The current state of the database is, so we are now at version 4.12. We are going to release soon a version 5, and I will talk about it. We have one of the big advantage of PR2 is to have a unified taxonomy. So we spend a lot of time curating the taxonomy, making sure that, for example, one genus doesn't belong to two different classes, because this happens all the time, actually. For example, if you go to GenBank, it's not very clean. So that's the advantage of one of the advantages of PR2. Right now, we have about 180 sequences that are annotated, and also we added last release, 6,000 samples sequence of plastine 60S that come from the phytoref database, which had been established by Johan Dossel, and he's in the audience, so I salute him here. Which are really useful because a lot of people, especially those working on phytoplankton, they try to combine data sets for 18S nuclear and 16S plastic. We have a quality control on the sequence, so we only take sequence larger than 500 base pair. All the sequences, by the way, are from GenBank. So they are not, we don't have private sequence or anything. It's just sequence from GenBank, either from 18th centuries or from genomes. Whoops, hold on, what happened? Sorry. Must have pressed too long. Yeah, sorry. We have metadata, which is really important actually, because this helps interpreting the data, the sequence. For example, you can see here the location of all the PR2 sequences that we extracted from the GenBank records. And finally, I will go back to this, but it's available under different forms in the web. And from late last year, November, we established a website so you can have information. This is not an interactive website in the sense that you cannot, for example, extract some given group, but you can have information about the sequence, news, and so forth, and how to use the database. So the database is structured as a database. Actually, it's under MySQL, and all the scripts that I use are written in R for importing, exporting, validating the data. The data are provided as files for metabarcoding and pipelines, and the most popular right now is Data2. Which now is going over Chime, over Mother, and so a lot of the downloads are for files compatible with Data2. We have also fast output for people that want really just to look at the sequence, and we have our package that not many people are using, but I will really encourage you to use, because it's really harnessing the power of PR2. It's very easy to install. I mean, if you are our person, but it's really easy to install and use. So we are providing some example actually on how to use it. And the beauty of it is that you can, for example, extract sequences from a given group and plot the metadata, analyze the metadata, and so forth. As I emphasize, the annotation is really the key part of PR2, how careful the annotation is. Some groups are not so well annotated, of course, some are better annotated. And this comes from all the contributors. For example, with the Dino5C, Solène Mordray, a couple of years ago, during our thesis, created a very good database for dinoflagellates. And those of you who know the dinoflagellate know that the taxonomy is changing all the time. So she really put a good effort into making the dinoflagellate taxonomy as up to date. Of course, it's going to evolve, so we need to. There will be a need at some point to renew this effort. And on the right, you can see all the groups that have been curated. Another way of annotation is using uCref pipeline, which was developed by Javier del Campo and some of his colleagues, where basically what you do is you start from a core set of sequences that you know for your group, then you go to GenBank, and using BLAST, you extract more sequences because we have only a subset of sequences. Then you can build a reference tree with these new sequences and refine the assignment. Especially, this is important for environmental sequences because, again, in GenBank, many of the environmental sequences are annotated as environmental sequences and no information taxonomy. While, as Mawash showed yesterday, there are some groups for which is there are very few culture or no culture, and it's important to have the taxonomy, the real taxonomy. And uCref works through workshops where people, 10 to 15, 20 people are coming There have been 3 workshops altogether. The last one was in Roscoff in 2018, focusing especially on diatoms and flagellates and green algae. And this annotation should at some point make it to peer 2. It takes time because it's not so easy. Actually, there is a lot of, everybody is very enthusiastic during the workshop, but after the enthusiasm goes back down and it takes some time to transfer. But for example, there was a very nice work which was done during one of the workshops for ciliates, and now the ciliates are quite very well annotated in PR2 using the UCREF. Next release should come soon, probably July, maybe August 2022. The big change is that we are going to go from 8 levels to 9 levels. Because it turns out that the root taxonomy of eukaryotes is changing all this time. So, for example, you may have heard about SAR, which were Straminopiles, Ardolata, and Rhizaria, and now we have SAR, which are Telonemia, has been added. So this higher level taxonomy is changing, and to accommodate this, we had to move to 9 levels with the very, very strong help of Javier del Campo, Laure Guillou, and all the team from Faktenburg. So this is going to be a big change, and especially also to accommodate the last taxonomy from the adult paper in 2019. We're going to add also new sequences. The sequence will not be in the core of PR2, but there are sequences that are from Silva or gene banks that have not been annotated. So they will be like in the back porch of PR2. And if you want to work on a given group, for example, to do the annotation, you could draw on this sequence and for later inclusion into PR2. So these are sequences from Silva, sequence from gene bank. And finally, we have been reannotating also some groups. For example, within the dinoflagellates, Ragier has reannotated the suicelial, which have, so the Symbiodinium, which have changed taxonomy recently, and with the creation of several new species corresponding to the different clades of Symbiodina. And for the forum, Raphaël Morar is providing us with a very, very nicely annotated database for forum. So who is using PR2? Right now, we have more than 380 papers citing PR2. And analyzing the abstract from this paper, you can see that the keywords is diversity. So this is used for diversity, but in many different fields, actually. And when you look at the papers, you find a lot of papers, of course, in marine, in freshwater, more and more in soil. And I know that Stefan Geisen is in the audience, so is one of these that are using PR2 for the soil, but also very strange, more strange, it's not strange, but more foreign application, like, for example, looking at the gutters of Paris, which are the eukaryotes in the gutters of Paris, and how can you use this to do some urban ecology, or even in forensics. So this paper here, which is using eukaryotes to make some post-mortem analysis. So we never thought that you would use PR2 for this kind of application, but obviously it's but also this kind of database can do many more analysis. So with Stefan Geisen and Fred Maé, we are currently doing analysis of all the primers that have been used for amplifying for metabarcodes of 18S. So we are building a database of primers, and then we are using PR2 to look at the specificity of these primers. And what you can see actually in the previous talk, it was said that, for example, the a low percentage of the excavata was amplified by V4. It was better with V9. In our database, we don't see this, but still, there are probably a lot of sequence from escamots that are amplified with V9, maybe not amplified with V4. I want to finish very briefly with another database that I've been building since about 5-6 years, which is a database of metabarcodes. There are a lot of metabarcoding data around in the for example, this is big expedition like Tara Oceans, Malaspina, the ocean sampling there also, but also many individual studies. And if you want to use this data to do some biogeography, for example, it's very hard because there are separate data sets. Usually they've been processed with different pipelines. There are different level of similarities. For example, in one of the talk, we heard that someone was using in the Korean study of the hypersaline using 97% similarity. You have different reference database. People are using Silva, some people are using Bringin, some people are using PR2. And most of the time, it's very hard to get the metadata. So my idea was to do a bit what we are doing with PR2 is to reprocess actually all the data set, but in a uniform way. So the way it works is that I'm downloading public data sets as raw sequences with the metadata. Then I use Data2, which provide amplicon sequence variant. And the beauty of Data2 is that different data sets can be merged. So for example, I can process Malaspina, I can process ocean sampling day, and then I can merge them together. If you do this with OTUs, it's impossible. You will never have overlap between the different data sets. You will find that's not the same OTUs, for example, representative sequence from OTUs that are used in one data set and the other. And then everything is in a MySQL database, which is a very efficient way to store this. And then this is processed with RScript. So right now, I have 32 data sets included and more are coming. I also have private data sets, people that we are our own data set. So we have for V4, we have the OSD, Malaspina, and region. So I try to download we have altogether this amounts to about 5,000 samples, mostly from oceanic waters, but I will try also to include other environments like soil environments, for example, and freshwater, and altogether to about 130,000 ASVs. So for example, just on the right, you can see for the photosynthetic, which genus has the most reads in this database. It's Procentrum. So that gives you an idea of the importance of the different genera. What you can do with this database, just one example is looking at Pelagophyceae, which is a very important group in oceanic waters. Everybody says it's very important, but there are very few data showing it, actually. And so with this database, we can begin to map different genera or species of Pelagophyceae, and this is work done by Ana Maria Cabello and people from Barcelona, Ramon Massana, Ramiro Logares. And what we are able to show is that in oceanic waters, you have Pelagomonas calceolata, which was the first described species by Bob Anderson in 1993, which is really dominant almost everywhere, but not everywhere. There's some region where it's not Pelagomonas. While Orococcus, which was initially isolated in the United States and doing coastal blooms in lagoons, is important actually in coastal waters, and there are more species of pelagophytes. So this is the kind of work we can do, and we are especially focusing on right now. I've been focusing on Arctic waters. Just so what is next? Almost wrapping up. I think the next frontier, as I said, is looking at the full RNA operon and annotating the full RNA operon. Continuing to annotate a specific group, and that's where I call upon you actually as contributors to try to contribute to PR2 and also to the UCREF pipeline and UCREF workshops. Getting more metadata, and what would be really also very interesting is to get some phenotypic data. There was a paper published last month that did this for bacteria. They did a fantastic job of looking through many papers, many databases, and they were able to assemble a database of phenotypes for bacteria. And I think that's the transition from using only the taxonomic information to using to go to functional analysis actually with this database. I would like just to acknowledge the people that funded or helped fund. No, this has never been funded directly, really. It was funded through projects where it was on the side, like Biomarks. The Moore Foundation funded directly UCREF, and of course CNRS Sorbonne University and NTU, where I'm visiting now. And finally, if you want, you're very welcome to browse through the website and where you will find more information. I'm done. Questions?
Thank you very much, Daniel. There is a question here on the chat by Fabian Burki that says, where can we access PR2 metadata?
First, there is a flat file, so which is available, where you have for each sequence you have all the metadata actually. So this is so you have the taxonomy of each sequence and also the metadata. But the best way is using the R package actually, because there you have access. It's the same basically, but you have you can filter, and so that's a way to access the metadata actually. There is nothing like a website. We're thinking about it actually, but I mean we don't believe it's much added value to access this information. But right now, the best way is using the R package.
Thank you. I don't know if there's any more questions there. In the meantime, I do have one, Daniel. Is there any intention of adding a layer of ecological roles or functional roles to the taxonomy in PR2? Even if it's
yeah, that was my last slide actually, where I was in the perspective here. Adding, I call it phenotypes, but okay, basically it's adding functional information. But this is far from being easy actually, because there has been a work, for example, in Roscoff, a student, PhD student that built a functional database for a few of the phytoplankton. He has like 2,000 entries. But after it needs to be verified. It needs also to be unified, because some people are using a vocabulary, some other people are using a different vocabulary. So it's in this work that I was mentioning on the bacteria, they spend a lot of time actually saying, okay, this term that these people are using is exactly the same than this term that these people are using. So that's a lot of work. That's the thesis work, basically.
Okay. Thank you. I don't see any more questions right now, but we can follow with the discussion afterwards in the parallel meeting if necessary.
Okay, I'll stop.
Thank you very much, Daniel. Adriana, I hand it to you now.
Okay, yeah, Karl, are you ready? You want to share your screen while I'm doing this long introduction for you?
A long introduction. Okay, I can share the screen. Thanks.
So it's a pleasure to introduce Karl. He's a phytoplankton ecologist. He's based in Newark. He has a really broad interest on phytoplankton from fresh water and also from marine water. He has a very, very long experience looking phytoplankton under the microscope, identifying them. He's also, I can say just from experience, he's one of those that you want to be on a boat with because you can run to him and ask what is this under the microscope, or have his help for anything that you need. He also has time to brew his beer and to win championship twice in New Zealand for his home brew. I need to taste next time. But today he's gonna talk about the results that he found, that he has from a recent voyage to the productive subtropical front, where he looked into diatom and parasitism. So Karl is with you.
Well, thank you very much for that introduction. I think it's longer than the talk I'm going to give. So as has been introduced, I'm going to discuss, this voyage was actually a little while ago, but it's one of the examples of parasitism that I've come across in the Chatham Rise subtropical convergence zone. I thought I'd just first of all remind us why parasitism is important and how it affects food web systems. Probably the primary way that it's affecting the composition of phytoplankton and its selectivity in this area is how it feeds through the food web, affecting things like grazing and having effects also on dissolved and particulate organic carbon. So what I'm trying to get at here is these sorts of organisms have big implications for our food web processes. The type of parasites I'm talking about today were the chytrids. This is a rhizophidium type, and this was an infection of diatoms that occurred on this Chatham Rise region, and this is a fungal infection for those of you who might not be familiar with it. So where was this parasitism observed? It was during a voyage to the subtropical Rise region, which is off the east coast of the South Island of New Zealand, for those who might not be familiar with this region. This is an area where we have subtropical waters meeting subantarctic waters over a frontal zone and an area of significant mixing. It's a high productive zone and it's an important fishery for New Zealand. So it's an important area to study. So how did we identify parasitism in this case? Well, this is a bit of a novel technique. We were running FlowCam imaging systems while we were going across the Rise. And we noticed within these images that we could pick up infections of the cells, and we could see sporangium and infected cells. So using these images and by doing multiple runs, we were able to estimate the rates of infection on different diatoms. And in this case, I put up a plots showing Thalassocyra, and this is showing a balloon plot of infection rates. And if you focus on the upper left-hand panel and you look at S10, you'll see that we had about a 15% infection rate at that site. As you can see, infection varied across the Rise. And to the lower left panel, you can see I've also included a plot of chlorophyll showing how chlorophyll was changing. So we had different biomasses. When we looked a bit deeper at this data, we found that we could see some relationships occurring. It occurred that as biomass increased in general in terms of chlorophyll and the abundance of particular species increased, we saw increasing infection rates. So abundance and biomass seems to parallel with infection. We also found that different host species had different levels of infection. So here I've shown an example of Thalassocyra, but species like Cattosaurus had higher rates of infection whereas species like Pseudonychia had lower rates of infection. We also found that some rare large diatoms were highly infected, and they may have declined before we arrived out there for this voyage. The final point I'd like to make in this short presentation is that production also declined with increasing infection rates. So in our measurements of production that we did during this voyage, we saw that when we had higher infection rates, we had lower levels of production in those sites. So just in summary, it seems like the flow cam could be a good way of at least initially picking this up. Obviously, we'd like to, if we see this event occurring again, get some good sort of metabarcoding data and other data to help confirm our findings. We also would like to do some more calcifluor type preparations to really examine the parasitism of these cells. We can see that this parasitism rates from this short study are linked to diatom speciation and size, and parasitism will affect productivity rates. So obviously this is an important area for New Zealand, so this sort of event could have significant impacts on the food web. So I think that's all I've got time for. Thanks very much. Please ask any questions.
Wow,
How am I doing
thank
for
you.
time? Okay.
So there will be time for questions during the coffee break.
Okay.
If I can ask you to unshare your screen. Thank you very much for a really nice short talk.
Thanks very much.
And Megan, could you please share your screen?
Yes.
So our next speaker is Megan Sørensen. Megan is a postdoc at the Stockholm University and at the same time Uppsala University, and she did her PhD in Sheffield with Mike Brockhurst, and she will talk about this fascinating symbiosis in Paramecium with Chlorella, and her work shows what I've been thinking a long time, that the term symbiosis is should be viewed only with context dependent, based on the term symbiosis is very context dependent, and sometimes it's not that useful, especially over long evolutionary timeframes. So Megan, please. Thank you.
Yeah. So thank you very much to the organizers for setting this up, inviting me to speak. As Philip said, I'll be talking about the rapid recovery of fitness in a partner-switched Paramecium Chlorella symbiosis. And so the background for this is that there's increasing evidence for the role of partner switching in a broad range of symbioses. And originally, we might have thought this was a rare process that doesn't occur that much. But as work continues, we see more and more that this is occurring in a broad range of relationships, and how crucial it can be that symbioses can switch their partners. And that this process is able to prevent the breakdown of symbiosis. So what it can do is help to prevent the loss of functionality, so that if a symbiont reduces to the point that it can no longer function, by switching in a new symbiont, the relationship can avoid extinction. And work has shown this in sort of spittlebugs and cicada. It's also been shown to occur when there's a mismatch with the environmental conditions, so that if environmental changes the context dependence of the relationship, then the partner may not be sort of able to function in the new conditions, but a new strain may provide functionality to the new conditions. And this has been seen in relationships such as Symbiodimium and coral. And therefore we see that by partner switching, rapid adaptation can occur far faster than genetic adaptation of the same symbiont would allow, meaning that it can lead to fast rates of niche expansion, which has been shown in lichen, and can generally provide a way for the partners to sort of have a very fast response time to any changes. The problem, however, with partner switching is that the novel host symbiont pairings often have low fitness to begin with because of their lack of shared evolutionary history. The question therefore becomes, how do these partner switch symbioses stabilize if they're so important for the long-term maintenance of these relationships? And are they able to rapidly increase their fitness in a timescale that allows this process to be realistic? So to answer these questions, I used a novel, initially non-beneficial Paramecium Chlorella symbiosis and studied the evolution of benefit, asking whether the novel pair could rapidly improve its fitness and whether this fitness was able to reach the ancestral levels, or whether genetic constraints would always mean that the novel relationship performed poorly, and by which mechanisms benefit was able to improve. But first, some background on the system. So Paramecium bursariae is a ciliate, and it houses a couple of hundred of the green algae Chlorella symbionts. This relationship is a classical photosymbiotic symbiosis by which the green algae photosynthesize and therefore provide fixed carbon in the form of maltose for the host. In exchange, they get organic nitrogen from the host, so the host has to maintain its feeding behavior. Usefully, this is mostly a facultative endosymbiosis, so though naturally the partners are almost always found together, in the lab we can separate the partners, grow them by themselves, and therefore switch strains between different partners. Interestingly, this symbiosis has had multiple independent origins, so that when we look at the Chlorella genetics, there are at least two clear biogeographical clades. One is known as the American-Japanese clade and the other is the European clade. But each of these symbiotic clades are more closely related to other free-living Chlorella species. So there's been convergence in this symbiosis. For the work I'm talking about today, I've used a strain from each of these two major clades, one from the European strain that will be referred to as the native species, and one from the American-Japanese clade, which will be referred to as the novel species. And because they are biogeographical clades, this means there's a very low chance that these strains have ever come into contact naturally beforehand. So previous work in the group I was in looked at whether this was in fact a mutualism or an exploitation, because when we see the exchange of nutrients, we often assume that this must mean there's mutual benefit. But in fact, things are often more complicated. And first, when we looked at host growth rate over the light gradient, we can see that in the dark, the symbiotic relationship is more costly than the non-symbiotic relationship, because the host has to maintain its symbionts without receiving any benefit. But as the light increases, the benefit of symbiosis for the host increases, and it becomes more beneficial to be symbiotic for the host. So we see this strong light dependency for the host. However, when we look at the symbiont, this is looking at photosynthetic efficiency of symbiotic Chlorella versus free-living Chlorella, we actually see that the symbiotic Chlorella are always less efficient than the free-living Chlorella, sort of suggesting that for the Chlorella, it would actually be more beneficial to be free-living than to be symbiotic. We think this is suggestive that this is actually a case of host exploitation, and that it is the host that is largely benefiting from this relationship, and not both partners in an equal manner. In line with this, we see that symbiont load regulation is more tightly aligned with the host benefit than the symbiont benefit. So if you look at Chlorella growth over a light gradient, you just see a linear relationship in which Chlorella increase with increasing light. But when we look at the number of symbionts per host cell, we see that there's a peak at low light, but then a decrease in the number of symbionts per host cell. This seems to align with the host perspective, because at the higher light levels, each individual symbiont is more efficient, meaning that the amount of carbon given to the host can be maintained with a fewer number of symbionts if you increase their efficiency. And therefore, the symbiont load regulation seems to maximize the benefit to cost ratio for the host only. And sort of this evidence collectively seems to imply that this is quite an asymmetrical relationship between host and symbiont. So setting up the experiment, the evolution experiment, I took a symbiotic relationship and cured the host of all its symbionts, creating so that it was completely free of the symbionts, and then reintroduced either its native symbiont back into the host or a novel symbiont that the host had never seen. The two different symbiont identities were confirmed by PCR, and then the evolution experiment was begun. This involved serial transfer over a 25-week period, roughly 50 host generations in a high light condition. Assays were performed at the start and end, and some throughout the experiment, and we looked therefore at whether these novel symbionts behaved in comparison to the native symbionts. First, we looked at the growth rate of the hosts, and here in the plot on the left, we can see the growth rate at the beginning, and here we see that the native, which is the blue line, has a much higher growth rate than the novel with the novel symbionts, which is the gray line, and that this is particularly apparent in the high light condition. However, by the end of experiment, we see that this difference has disappeared, and that the growth rate of the two symbiotic units are now indistinguishable, and that the novel symbiosis essentially looks like it has managed to increase its growth rate to completely match that of the native symbiosis. We confirm this further by looking at the fitness. So this was done using direct competition fitness assays. So the symbiotic cells were directly competed with their symbiotic free ancestor, starting off with 50-50 populations, and then the proportions were studied over a fixed period of time. By matching these, it's possible to calculate a selection rate that then can therefore be a sort of quantification of their fitness that can be compared at different time points. Here we saw the same result, so that at the start of the experiment, the plot on the left, we see that the native symbiont has a much higher selection rate than the novel symbiont at the high light level, but that this difference has disappeared by the end of the experiment, and that the low fitness has been completely ameliorated, and that the selection rates are now matched at the high light level between both the novel and the native symbiont. Following on from this, we looked at the symbiont load regulation in the two symbioses, and here we see that at the start of the experiment, the native symbiont load is much higher at the higher light condition than the novel symbiont load, but that this is actually reversed by the end of the experiment. So that it looks like in the novel symbiont has been upregulated over the course of the experiment. And it's perhaps simply the increased number of symbionts that may have led to a higher host growth rate, because more symbionts will provide more carbon for the host. What's interesting is that the native pair also appears to have decreased its symbiont load, perhaps suggesting that the benefit per symbiont has increased, because we've seen that the growth rate and fitness didn't decrease. So it's able to maintain these same levels of output, but with a fewer number of symbionts, which is perhaps because of an increase in efficiency. Following on from these phenotypic measurements, we wanted to investigate the mechanisms of adaptation at a metabolic level, which would allow us to distinguish between host versus symbiont contributions. We used untargeted mass spectrometry to analyze the host and symbiont metabolism separately, and compared these again between the start and the end of the experiment. So looking at the Paramecium metabolism, so these are trajectories in PCA space from the start points, which are the darker points where the lines begin, to the end of the experiment, which are where the arrows end. Sorry. And we can see that on the plot on the left is PCA1 versus PCA2, whereas the plot on the right is PCA2 versus PCA3. And in the Paramecium fraction, there is a strong signal of convergence between the two starting points. So at the start, the hosts with different symbionts have slightly different profiles. By the end of the experiment, these profiles have merged and they share the same PCA space. This final space that they end up in is associated actually with a decrease in a number of metabolites, including metabolites of central metabolism, antioxidants, and lipids. We also looked at the abundance of these metabolites specifically. So this heat map shows some of these key metabolites that the PCA space was associated with. And here, the two lines that have these sort of the dark red and high abundances, these are our starting samples. And the ones in between that have a much lower number of much lower abundance of a large number of metabolites are our endpoints. And we can see that there is a large number of them that really have a strong decrease over the course of the experiment. And in particular, this does involve some antioxidants, which are very important in photosynthetic relationships. One of the things that did actually increase, though, in some of the samples is a number of algae cell wall degradation products, which potentially suggest that some of the algae are being digested later on in the experiment. When we look at this in the Chlorella metabolism, we see a more complicated picture in that two different trajectories occur over the course of the experiment. One is followed by all the native symbionts, so these are the ones in blue, and two of the novel symbionts, whereas the remaining four novel symbionts go in a different direction. These four that go in a different direction are associated again with a decrease in abundance of key metabolites, whereas the novel symbionts and those two of whereas the native symbionts and those two that go off together in a direction are associated with an increase in lipids, carbohydrates, chlorophyll, and carotenoids. So when we again look at these abundances of the metabolites in the heat map, we see that sort of this section of the map, which is the native symbionts and those two replicates of the novel symbionts, have these higher abundances of metabolites, chlorophyll, lipids, and some carbohydrates, whereas the four that go in a different trajectory have these lower abundances. We also see that there is some increase in the carotenoids and secondary metabolites associated with oxidative stress. And therefore, when we compare the two, we do see sort of some links between the Paramecium and Chlorella metabolism. So in the Paramecium, we see all that decrease in antioxidants, where in the Chlorella, this is matched with some samples in an increase in antioxidants. So it appears that the Chlorella is taking over the the oxidative stress response for both partners. In the Paramecium, we see a convergence of the host, regardless of what symbiont they have metabolically, whereas in the Chlorella, we see these two different trajectories arising. The convergence in the Paramecium is associated in a decrease in aspects of central metabolism, and in the Chlorella, the one trajectory is also associated in a decrease in certain metabolites. However, the second trajectory that's taken by the native symbionts and two of the novel was associated with an increased investment in photosynthesis and photoprotection. And this could account for an increase in carbon transfer and ability to photoprotect, would perhaps provide the mechanism for an increase in benefit per symbiont, which may explain that decrease in symbiont load that we saw in the native symbionts. This changing in symbiotic load would also be linked to the increase in algae cell wall degradation products that we saw in the hosts, as it is digestion is one of the mechanisms by the host can change its symbiont number. So overall, metabolically, we see that the symbionts take over the oxidative stress response, that the symbionts have two trajectories, whereas the host converges on one, and that appears to be that metabolism and symbiont load are linked, and that through that symbiotic load provides a way of regulating and responding to the different metabolic trajectories of the symbiote. So in conclusion, we have seen a really surprising rapid evolution of benefit in this novel host-symbiotic pairing, and that it only took 50 host generations for this novel pair to acquire the same fitness as the native pair. This evolution of benefit appeared to occur through changes in symbiotic load and metabolism and the link between the two. And we think that it's the fact that these are both regulatory mechanisms, not requiring genetic changes, that allows such a fast response and does provide a mechanism by which symbioses can respond quickly to the pair that they find themselves in. Overall, we have the hypothesis that there was two evolutionary strategies here. One was to have fewer, more efficient symbionts, and the other was to have more less efficient symbionts. And therefore, it was this matter of linking both symbiont load and metabolism that led to the evolution of a higher fitness in this novel pairing. So returning to the original questions, we think that this work has shown that the rapid evolution of benefit is possible, showing following a partner switching in symbiotic relationships. And that this occurred at a surprisingly fast timescale, meaning that the initial low fitness period can be overcome surprisingly quickly, especially when regulatory mechanisms such as symbiont load and metabolism are used. This means that partner switching can occur with a broader range of partners than initial compatibility tests would reveal. So if we're talking about partner compatibility, we need to be aware of what timescale as well as what context we're testing these partners in, and that they are very dependent on both. But that overall, that the fast adaptation within these symbiotic relationships does mean that low fitness can be overcome, and that partner switching can occur within a sort of reasonable time frame, that it can lead to, that it can stabilize and help maintain symbiotic relationships in the long term. And with that, I would like to just thank my PhD supervisors, Mike Brockhurst, Duncan Cameron, and Jamie Wood, as well as the group that I was in for my PhD work. So this happened at the University of Sheffield. And that is it.
Thank you, Megan, for a really nice talk. We have one question in the chat. So Rebecca Case is asking, what light level did you grow them under between T0 and T25?
Yeah, so the evolution experiment happened at a high light level. So this is the 50 microeinsteins, which is, so the highest light level in the graphs.
So I have a question. Is there any molecular dating available for Paramecium or Chlorella? Do we have any idea how old or new the symbiosis is, and how many possible replacements could occur since then, since to get to the native state only takes 50 generations?
Yeah, so I'm not aware there having been much molecular dating work, which would be very nice to know for especially the different, and whether the clades have, the two different clades have similar or dissimilar. But I don't think, unfortunately, that's been done. But yeah, it does, the fact that it can happen at such a fast rate does really imply that you can have a large number of sort of serial switches and not really be able to sort of pick up on it very quickly. So yeah, I think it does lead to the fact that there's probably a lot more switching going on that we're aware of, and that they're quite fluid relationships as once that it is established. I mean, so you can't swap them into Paramecium that haven't had Chlorella before. That's much harder. But once the host symbiote relationship is sort of established, then it seems to be able to switch fairly easily.
Thank you. We have a follow-up question from Rebecca Case. So do you think this is evolution if it's genetic regulation? I would suggest this is phenotypic plasticity.
Yeah, I mean, I think that's a sort of interesting point, and whether it is sort of matched between the two. But I think it's more stable than just phenotypic plasticity, but I think it depends on how you use those words, because it's not something that happened within, it's not something that can happen within a generation. The original generation could not adapt its phenotype and regulate its symbiont load or metabolism immediately to the new symbiont that's put in. So it has to happen over a multi-generational effect. It's just that it's able to do so without genetic mutation, if that makes sense. So I think it's sort of blurring the edges between, well, it depends quite how we define sort of phenotypic plasticity and adaptation, but it can't occur within a generation. So it's not quite just changing your phenotype.
Yeah, so Rebecca is thinking about intergenerational gene regulation. Yeah, this is for maybe for discussion in the parallel meeting. And we have one more question by Iona Brandstrom. So do you expect to find Paramecium hosts with more than one Chlorella genotype?
Yeah, so I think that would be quite interesting. It's not, there has been there's been one study that seemed to find that you could find multiple symbionts within the same one, but it doesn't seem to happen very often. So I think for the most part, it is just one, and which would, in terms of sort of symbiotic stability, it helps a lot if you're not having competition between symbionts within the same host cell. But it does seem to occasionally occur, but whether that's a sort of transitional period and that it's always that one eventually takes over, or whether it's actually possible to stabilize with two, I don't think we have enough data for. But it would be really fun to run experiments that actually competed them within single host cells.
So there are no more questions. So thank you, Megan. And I will ask Drahomíra to share her screen for the next lightning talk. So Drahomíra is a research scientist at the Institute of Parasitology of the Czech Academy of Sciences and Julius Lukács Lab. And she will tell us more today about this massive community effort to build genetic tools for marine protists. So Drahomíra, please go ahead.
Mm-hmm. So hello everybody. Thank you very much, Philip, for inviting me, and all the organizers for organizing such a nice meeting. Today I would like to talk, as Philip mentioned already, about the huge paper that we published last month in Nature Methods. Its name is Genetic Tools Development in Marine Protists, Emerging Model Organisms for Experimental Cell Biology. And as you can see here, it was really a big project with 113 collaborators from 53 laboratories and 14 countries all over the world. And me and my supervisor, Julius Lukács, were the main coordinators of this study. The goal was to try to establish as many marine species as possible to be a model organism, to be able to study their genes, as till now not so many of them were available, or very little were available. So we studied altogether 39 protists from 7 main eukaryotic groups. So from all of them except metamonads and amebozoans. And since I have just 5 minutes, I will show you the main outcome on this slide. So we were able to establish like 22 protists and establish genetic tools for them. this is a busy slide, but we managed it for 4 archaeoplastids. So you can see the colors, the same species, and they are listed always the species that belongs to the group. So for example, for archaeoplastids, as I said, we mentioned it for 4 species. They are labeled in yellow for DNA delivered and expressed. Construct for DNA delivered only, it's in gray, and for not achieved yet, it's in blue. The other group, there is 1 haptophyton, 1 rhizarian that we managed to do the genetic tools for it, 6 travenopiles, 5 alveoloids, and 3 discobands, and 2 opistocons. We didn't summarize not only what worked, but also for the 17 labeled in blue, we described what we were able to find out. For example, the resistant markers, so the other groups can also continue on this study. So how to make a model organism? We established how to make an transformation protocol. So first of all, you need to describe the organism you are working on. Ideally, grow it in maximum cell density in the lab, without any bacteria, if possible. To check the autofluorescence to be sure that it will not interfere with potentially introduced fluorescent markers. And of course, it's necessary to sequence the genome. If possible, the cryopreservation is also a great to be done. Then 3 things has to be fulfilled at the same time, otherwise it will not work. So it's great to have the resistant markers, the construct suitable for to be expressed in the species, and transformation method. Which is sometimes very difficult to find at the same time, and then it's a problem, and this whole transformation simply doesn't work. And then if it works and we receive the clones, the transformants can be verified at different level, at the level of the DNA, RNA, and translation by Western blot analysis, epifluorescent microscopy, and also protein localization. So we find out that there is no universally applicable protocol. And for every species, we basically established a unique protocol. There is a huge table in the paper where we list for every species the particular conditions. For the ones that it worked, we were able to verify the transformation of these species by epifluorescent microscopy. So for example, here you can compare the wild type fluorescence with the transformed cells. Here the red or green fluorescent proteins are expressed. The transformation in the other species was also confirmed by luminescence or fluorescence, or by RT-PCR, reverse transcription PCR, Western blot analysis, and also by CRISPR-Cas9, based on the construct introduced into the cells. The outcome of the paper, or the summary, is making a transformation roadmap. And here we describe how to proceed when someone wants to try to do the model organism. So how to establish, how to construct the vector, and which other transformation approaches are possible. And as I said, I could not say all the details, so the paper is available online. It's open access, so for more details, you can either send me an email or read it. And I would like, first of all, I would like to thank all the co-authors for great collaboration on this project. And we also would like to thank Gordon and Betty Moore Foundation, especially their experimental model system program of their marine microbiology initiative, for support, and also Protocols.io for storing all our protocols. And I would like to thank you for your attention. Thank you. So if you have any questions, we can meet in the...
Thank you, Drahomíra. Adriana, could you please take over? Could you please?
Yeah.
Thank you.
Mariana, are you there?
Yes, I will share my
Okay.
screen.
Okay, so it's a pleasure to introduce Mariana. Mariana is a PhD student at in the plankton group at Roscoff at the Station Biologique. She's studying the microbiome of the earth. She's graduated in Brazil. She's from Brazil also, as a microbiologist, and also in Brazil she became a chef, where she knows how to do very nice desserts using a very special type of liquor from Brazil. So Mariana is with you.
Thank you, Adriana, for your introduction. And it's a pleasure to be here. So I will present you some results about the microbiome of Emiliania huxleyi, and we chose this model besides by its ecological relevance, also because E. huxleyi is easily cultivable. So we have many strains available in Roscoff that were isolated from everywhere in the world. So the main objectives that I will focus here are three. So the first one, to investigate the main drivers of microbiome composition in huxleyi cultures, to compare its microbiome composition to that of other microalgae, and to investigate if the interactions found in cultures are also present in the ocean. So I have two main data sets. The first one, where we selected 77 strains, 62 from Emiliania huxleyi that came from different places in the world and have different time from isolation, and 15 from Diatom oceanica, which are closely related to E. huxleyi, so to understand the specificities and the drivers of the microbiome composition. And the second one, where we selected other microalgae that came from the same samples as E. huxleyi. So besides E. huxleyi, we had other aptophytes, Mysozoa, Ochropta, Chloropta, and Bacillariopta. So from the xenic cultures, the cultures with the bacterial community associated, we use metabarcoding analysis of the V4, V5 regions, and I used the DADA2 pipeline to obtain the amplicon sequence variance table for the community analysis, and then we started to look for some ASVs in the global ocean data of Tara Oceans expeditions. So first, we didn't find any relationship between the microbiomes and the variables that we used for selection. So here in this ordination, I highlight the microbiomes of Emiliania huxleyi and the Phycapsa, and we don't have a clear pattern between them. What we observed is that we had true Marinobacter ASVs that were present in all the cultures and in variable abundance. So in this heat map, I consider all the ASVs with more than 5% of the reads in each microbiome. And we can clearly see that we had three clusters based, one dominated by ASV3, another one dominated by ASV1, and the third one that was not dominated by these ASVs. And when we faced these results, we were intrigued if this could be a bias of the culture conditions that would be selecting a homogeneous microbiome even between different species. So to further investigate that, we had then other microalgal cultures that came from 6 sampling cruises, and what is interesting here is that they came from the same samples, the same water samples in each cruise, and we had also the environmental parameters to correlate with this data. It's what I show in this distance-based RDA. So here we have all the microbiomes from the 6 sampling cruises, and I included in this model temperature, salinity, phosphate, and total chlorophyll A. And what we see is that this model explained 15% of the variance in the composition of these microbiomes, showing that even after years in co-cultivation, the microbiomes still are correlated with the environmental parameters of the sites of isolation. What we found also is that, again, ASV1 was present and in all the cultures, so which seems that this ASV is able to colonize all the kind of microalgal cultures. And ASV3 appeared, but now restricted to few E. huxleyi and Phycapsa cultures, which are both aptophytes. So what we are doing now is to look specifically for some ASVs in the global ocean data of the Tara expeditions. So I would just briefly show preliminary results, for example, for ASV1. So here in the crosses, we have all the Tara ocean stations, and the shapes and the colors corresponds to the size fractions that of the metabarcodes data that we checked. So the circles from 0.8 to 5, the triangles 5 to 20, and the squares 20 to 180 microns. And what we see first is that this ASV again appear in almost all the stations of that were analyzed, which kind of confirms what we found, that this ASV is able to colonize all kind of many kind of environments, and they are mostly abundant in the larger size fractions, which could indicate an interaction with phytoplankton organisms. But what we are doing now is to check starting to investigate the interactions between these ASVs and other organisms in the global ocean data. So I would like to conclude. So we found the different Marinobacter ASVs dominating the microbiome of huxleyi culture. We found also that the microbiome composition in culture is correlated with physicochemical conditions of the time and place of isolation, even year after years in cultivation, and also that this association of culture-based microbiome studies and global ocean data will help us to unravel important microalgae bacteria interactions. So I would like to thanks again to the organization of the event for giving us this opportunity to share our research, and also to my advisors and to the team in Roscoff involved in this work. So thank you very much.
Thank you, Mariana. Andres, can you please share those slides? So before we go into the coffee breaks, I just wanted to mention a few things from the survey. Andres, can you go a few slides back? So I just wanted to highlight one result from the survey of the previous meeting. So for questions about should electronic registration be added to face-to-face meetings, the answer was almost all participants answered yes, 100 something. And would you register for a meeting in this way? Again, mostly yes. So there seems to be a big demand for at least partially electronic meetings in the field. Of course, this is biased by the people who replied to the survey, so participants of the previous meeting. But one interesting thing is that about half of the people don't think that electronic meetings should completely replace some face-to-face meetings. For example, alternating years for society meetings. So the take-home message from this survey is that most people seem to favor a hybrid model where some talks are live through Zoom, or some participants can attend only electronically, not only because of carbon footprint of the meetings, but also because other reasons like childcare. Some meetings provide childcare, but it's not always ideal. For example, with small children, or if both parents are scientists. So there's a big demand for this kind of a hybrid meeting with some part of the meeting being electronic. So I feel like a take-home message for future meeting organizers in the field is that they should implement electronic registration what I think is that the talks could be entirely online, and actually when we arrived to the venue, we could focus on the networking or field work or the actual in-person interactions. So if there are any organizers of Protistology meetings, this is something you should know, and we are happy to help with the electronic part. So now we have some experience, and I'm sure the previous meeting organizers would be happy to help as well. Andres, can you move one slide? I forgot to mention that we'll probably also send a survey to the participants of this meeting, mostly focusing on the Asia and Oceania region, asking questions about if you're interested in a meeting like this next year, or at least partially having this meeting combined with another meeting if the meeting is in North America or Europe. One thing that was also interesting from the survey is that some people would prefer to have a poster session, but some people don't. So it was not entirely clear from the survey. And we did not manage to implement a poster session during this meeting, but there's an alternative Adriana already mentioned. So please feel free to register for the poster session. They are kind enough to have a venue also for the Asia and Oceania time zone. Andres, can we go one more slide? And before we go to the coffee breaks, I just wanted to say thank you to all the organizers, because this was a massive effort. I would like to thank Adriana and Maggie, who helped a lot with all the organizing, and Andres. So this was the team in our time zone, and also Laura and Mark, who did a great job as a bridge with the European time zone. Also, I'd like to thank all the speakers who did amazing job being on time, presenting amazing talks, and of course all the participants, because without the participants there would be no meeting. I think yesterday we had around 160 unique participants. I'm not sure about the number for today. And we're fairly happy with the number of participants. So the purpose of this meeting was slightly different from the previous meeting. There are probably not that many protistologists in the region. So thank you for joining